vibrant greenery in our parks to the nourishing crops on our tables. They bring life and beauty to every corner of our lives. Beyond the practical benefits, plants also nourish our spirits, offering peace, joy, and deep connection to nature. Whether it's a shade of a tree or the bloom of a flower, the beauty of plants is truly everywhere. Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. Welcome back to my YouTube channel, where we're continuing my series on plant physiology. Today we're diving deep into the inner workings of plants, specifically how they store and metabolize carbon. From photosynthesis to the complex biochemistry inside plant cells, we'll uncover how plants fuel their growth and ultimately store that energy. Stick around to find out how this process is crucial, not just for plants, but for all life on Earth. Lesson 10, Carbon Metabolism and Storage. So let's get started with the basics. Plants capture carbon dioxide from the air and convert carbon dioxide into sugars through photosynthesis, as shown here in the map of central primary metabolism. Plants also have secondary metabolism, but this is used largely for defensive purposes. Photosynthesis happens in the chloroplasts inside their cells. Think of chloroplasts like little factories. They use energy from sunlight, carbon dioxide, and water to create triose phosphates, which gets converted to sugars, including glucose. Glucose is the primary molecule that plants use to store carbon in the form of carbohydrates, like sucrose and starch. But how does this stored carbon get used for growth and other functions? Once triose phosphate is produced, it can take different pathways inside the plant. Some of it is used immediately for energy through respiration, where glucose is broken down to produce ATP. This powers everything from cell division to the growth of leaves, roots, and fruits. But plants don't use all their glucose right away. They store the excess carbon that is produced. Now here's where things get interesting. Carbon storage in plants primarily happens in two forms: starch and sugars. They can be stored in various tissues and organs. Starch is stored in the chloroplast during the day and broken down at night when there's no sunlight for photosynthesis. It acts like a battery, releasing energy for nighttime growth. Plants also transport sugar to other parts of the plant through the phloem, particularly to the sink organs like growing apexes, roots, fruits, and seeds. This is known as sink demand, and sink demand controls whether sugar is directed to the phloem or the excess triose phosphate is directed to starch synthesis in the chloroplast. For long-term storage, excess carbon is stored as starch in organs like roots, trunks, and tubers. Think potatoes. Carbon is also crucial for building complex carbohydrates like cellulose, which make up plant cell walls. Cell walls provide structure and support. So in a way, carbon storage also directly contributes to the physical growth and stability of plants. What happens when a plant needs that stored energy, especially during periods when they aren't photosynthesizing as much during winter or drought? This is where metabolism kicks in through a process called glycolysis. Plants convert stored carbon back into glucose, which can be used for respiration. This keeps the plants alive and growing, even when external conditions are ideal. Here's what we will cover today. In our photosynthesis lecture before, we focused on the production of triose phosphate in the chloroplast. This is represented here with an orange circle around it. Triose phosphate is utilized in making sugars. First, we will talk about sugar chemistry and the interconversion of sugars. Second, we will cover sucrose synthesis. Third, we will cover the production of starch and starch catabolism, or degradation, or breakdown if you want. Fourth, we will discuss how triose phosphates can be converted to organic acids, which can be used in storage in the vacuoles of cells, but also transported to the mitochondria, where its energy can be converted into the production of ATP energy. The organic acids are moved into the citric acid cycle, which occurs in the mitochondria, which produces reductive compounds like NADH and FADH2. These reductive compounds transport electrons to the mitochondrial inner membranes. I will give more detail on the electron transport and production of ATP a little later in this lecture. Finally, we will discuss the synthesis of cellulose and the complexity of building cell walls. This impacts cell elongation and growth, which will be covered in the next lecture. Now let's go a little bit deeper and more specific. Let's talk biochemistry inside plant cells. The metabolism of carbon is tightly regulated by enzymes. These enzymes control whether carbon gets stored to starch or is used for immediate energy. It's a delicate balance that ensures the plant has enough energy to fuel growth while also building reserves for tough times. Before we go any further, though, let's look a little bit at some of the terminology for sugar chemistry. You can see a glucose molecule up on the upper left-hand corner of the slide. It is shown as a hexose, a 6-carbon sugar, and in ring form. It is a monosaccharide, a single sugar molecule. Next to it, we can see sucrose. It is a disaccharide. It is made up of 2 sugar units put together. In this case, sucrose is made up of a glucose molecule and a fructose molecule. On the right-hand side is a polysaccharide. Polysaccharides are made up of multiple sugar units greater than 40 units. In this case, we can see starch with between 300 and 600 units of glucose. So that's right, starch is simply a long chain of glucose molecules. Oligosaccharides, not shown here in this slide, are sugar molecules in length between 4 and 40 sugar units. So it's only when they become larger that we call them polysaccharides. Now we need to speak a little bit more about the bonds on a glucose molecule, because there's an important distinction between starch and cellulose, which is also a glucose polysaccharide. So if we look at the structure of a disaccharide consisting of 2 glucose molecules in the lower left of the slide, the type of bond that links them is called an alpha-1,4 glycosidic bond. That is, the first carbon of the glucose molecule on the left is bound to the 4th carbon on the glucose molecule on the right. It is an alpha bond because the bond is formed when the hydroxyl group on the carbon 1 of the first glucose is below the ring plane. It is a beta bond when the bond is formed when the hydroxyl group on the carbon 1 is above the ring plane, as shown here in the lactose molecule on the right lower section of the slide. This has an important impact on these bonds. In this figure, you can see a comparison of the starch and cellulose molecules. Both of the molecules, starch and cellulose, are made up of glucose molecules. However, the major differences between them is the glycosidic linkage. The glycosidic linkage is an alpha-1,4 linkage for starch and a beta-1,4 linkage for cellulose. The alpha-1,4 linkage of starch is readily degraded, but the beta-1,4 linkage is much more difficult to cleave, and it confers tensile stiffness that enables the cellulose polysaccharide to have the strength of steel in the cellulose microfibrils of the cell wall. So let's look in more detail at this conversion or interconversion of hexose phosphates. First, we go back to where we began with the triose phosphate produced in the chloroplast. These triose phosphates are transported out of the chloroplast. This is regulated very tightly by multiple enzymes in and outside of the chloroplast. We won't go into this tightly regulated control here, but just be aware that this is well studied and understood. In the cytoplasm, triose phosphate is converted to fructose 1,6 bisphosphate by an aldolase enzyme. This is then converted to fructose 6-phosphate, and then to glucose 6-phosphate, and then to glucose 1-phosphate. These reactions can be reversed to provide a plentiful pool of hexose molecules. This leads us to the second pathway. Sucrose synthesis utilizes one of these hexose phosphates, glucose 1-phosphate. Glucose 1-phosphate, along with UTP, is converted to UDP glucose. Sucrose 6-phosphate is produced from UDP glucose and a fructose 6-phosphate. Sucrose is produced once the phosphate group is removed by the sucrose phosphate phosphatase. Okay, let's move on to pathway 3, which is the formation of starch. It starts with the conversion of glucose 1-phosphate and ATP into ADP glucose. ADP glucose can then be attached to the end of an already existing starch granule. This is known as the elongation step, where a glucose is attached to a primer. This process can be repeated many times by a starch synthase enzyme to produce a long linear chain of glucose molecules using alpha-1,4 glycosidic linkages. This linear form of starch is known as amylose, and this type of starch represents about 30% of the total amount of starch. It turns out that starch's structure is much more complicated than singular chains of amylose. It has the ability to form branches utilizing a branching enzyme. The branching enzyme catalyzes a reaction to make an alpha-1,6 linkage between the two glucose molecules. This branched form of starch is known as amylopectin. In this slide, you can see an overall schematic of starch synthesis, with the elongation of amylose molecules and the formation of amylopectin by branching enzymes. This structure can vary considerably, with large numbers of branches randomly formed. Amylopectin can make up between 70 and 80% of the starch, and varies depending on the plant species and storage organ. And in this slide, you can actually see the well-developed starch granules in the chloroplast in this electron micrograph. This is what a chloroplast would look like at the end of the day as the starch continues to grow during the light of day. Once night comes, photosynthesis stops and starch no longer accumulates. In the night, this starch is utilized for normal cellular energy and functions. These are finely tuned processes so that the starch is used up at the end of the night and the chloroplast can start to synthesize starch again the next morning. So let's see how this starch degradation occurs in a leaf. This figure shows a chloroplast with starch granules in it, in the process of degrading or metabolizing or catabolizing a starch granule. ATP is required as it is necessary for the starch to be phosphorylated by kinases before it can be broken down by debranching enzymes. The resulting linear chains of starch can then be broken down into glucose molecules by beta amylase and D enzyme. Glucose 1-phosphate can also be directly synthesized. These glucose forms can then be interconverted to reform triose phosphates in both the chloroplast and the cytoplasm. Clearly, sugar metabolism is very complex. Whether it's night or day, plant cells need energy in the form of ATP. In this slide, we can see the summary of the fourth pathway, respiration. We can see portions of the map showing the conversion of triose phosphates into organic acids. These organic acids are subsequently transported across the membranes of the mitochondria, where they enter the citric acid cycle. In this process, important electron transfer molecules or reductants are formed as NADH and FADH2. These electrons are transported to the inner membranes in the mitochondria, where ATP is produced. The other parts of this figure show actual structures of a mitochondrion with an electron micrograph cross sections. You can see that a mitochondrion is made up of a series of membranes. There's an outer membrane, an inner membrane, and the matrix within, which are all important parts of the compartmentation within the mitochondria. This compartmentation is necessary to enable the mitochondrion to produce energy, as documented in the figure on the right. This process was first described accurately by Peter Mitchell with the chemiosmotic hypothesis for the production of ATP. He was awarded the Nobel Prize for his insights. This process is very similar between both plants and animals. This process starts with transfer of electrons from NADH and FADH2 through the electron transport chain in the inner membranes. This causes charge separation and a proton gradient across the membranes. Basically, this is very similar to the mechanism of charge separation by the electron transport chain in the chloroplast for photosynthesis, as we discussed in an earlier lecture. The separation of charge and the gradient of protons is utilized to make ATP through an ATP synthase. So let's look at the last pathway, cellulose synthesis, and the development of cell walls. First of all, cell walls vary a lot. You can see in this slide that the thickness and the composition of the cell walls are quite different amongst the different cells in this cross section. The fact that you see red walls here is the result of a dye staining for lignin, which is another complex molecule in the cell wall. We will not go into very much here, but the walls are also made up of hemicelluloses and pectin molecules. Here we will simply talk about cellulose, which is the main component of cell walls, making up about 50% of the total wall composition. Cellulose is the most abundant polysaccharide on the planet and is the component of paper that we use to write on. So let's look a little bit more in detail regarding the structures of cell walls. In this figure, we see a freeze fracture of some onion cell walls. We can see that there are different layers of cell wall. This is a transverse section where we can actually see these little dimples or holes through the cell wall. And these are the plasmodesmatal connections that are going between the cells. So what can we learn from these cell layers? How are they created? We can look a little bit more closely at the structure of the cell wall if we remove the hemicelluloses and pectins from the cell wall using various chemicals. What remains then is the cellulose in the wall. This figure demonstrates what this would look like. If we zoom in, we can see that there are fine strands of cellulose entwined and enmeshed throughout the wall. This is a transverse view of the wall. If we look through a cross-section of the wall, we can see the layers of the cell wall. In this view, we can see at least 8 or more layers. In this figure, we can see a model of how the cell wall synthesis is dependent upon several processes. One component uses Golgi vesicles to deliver precursors to the cell wall by a process known as exocytosis. These secretory vesicles merge with the plasma membrane and release the contents of those vesicles out into the cell wall space. The components delivered are largely made up of the hemicelluloses and pectins that are incorporated into the cell wall. By some manner, they get integrated into the wall structure. Also shown here is a cellulose synthase complex. This enzyme complex is responsible for the synthesis of cellulose microfibrils. You can see a strand of cellulose extending out and into and through the wall. It becomes integrated with the hemicelluloses and pectin molecules to make an integrated cell wall fabric. These cellulose microfibrils are even more complex, representing multiple strands of cellulose. If we look at the cross-section of the cellulose microfibril, we will see that the interior part is crystalline and highly organized. The external surface cellulose strands are less ordered. The cellulose molecules are held together by hydrogen bonds between and within the adjacent chains, giving the structure more rigidity. These hydrogen bonds also form between the hemicelluloses and are an important resistance to cell wall expansion as the cell grows. In this electron micrograph of a freeze fracture slice along the outer surface of the plasma membrane, we can see in the blowup a cellulose synthase complex that consists of 6 separate subunits of cellulose synthase. If one looks closely across the surface in the rest of this picture, you can see many complexes all along the surface of the plasma membrane. So there are a large number of these complexes in the membrane. In this freeze fracture of the inner surface of the plasma membrane, one can see that these microfibrils extend out through the cell wall. On the inside, there appears to be a thickened rail, which is part of the cytoskeleton. There is evidence to suggest that the cytoskeleton directs movement of the cellulose synthase complexes. In this figure, we can see how they align with each other. The orientation of these microfibrils along the surface can impact cell shape and elongation of the cell. So let's look at this process in a little more detail. We know, in fact, that these rosettes on the electron micrograph are the cellulose synthase complex, and they consist of 6 subunits, each of which consists of 6 cellulose synthases. Thus, a complex is synthesizing 36 chains of cellulose at one time into a cellulose microfibril. Okay, so the cell wall is quite complex, and the structure of these cellulose synthases is complex as well. Let's look at a model of what we think is going on here. We can see the structure of a single subunit of a cellulose synthase complex. In the center of the subunit is another enzyme called sucrose synthase. This sucrose synthase catabolizes sucrose, transferring the glucose component of the sugar to the cellulose synthase in the form of UDP glucose. The fructose molecule moves off into the cytoplasm. This enzyme belongs to the sucrose synthase family of enzymes, but in fact cleaves sucrose rather than synthesizing it. The UDP glucose is utilized by the cellulose synthase and attaches a glucose to the end of the cellulose chain as we previously described. However, there's no give in the cell wall because of the hydrogen bonding and interlocking of the other cell wall components. So in order to make room for an attachment to the end of glucose, the cellulose subunit is pushed through the fluid plasma membrane by the force of the extension. And as I said earlier, the direction of this movement is guided by the cytoskeleton on the inside of the membrane. So we can summarize here that we talked about 5 different pathways in which carbon is metabolized, utilizing triose phosphate from photosynthesis. One is the formation of other hexoses for other metabolic processes. 2 is to make sucrose that can be used to transport through the phloem. 3 is to make starch. When sucrose is no longer needed, the plant senses that and switches its metabolism to make starch. In the 4th pathway, triose phosphates get shuttled into mitochondria via organic acids and then are utilized in respiration to make ATP and energy for the cell. The 5th carbon pathway is the formation of cellulose as I described here. In the next lecture, I will talk about how the structure of cellulose is important for the cell shape and the ability of the plant cell to expand, and on a larger scale of the entire plant. How does a plant grow? How is it coordinated, particularly between roots and shoots? What are the control points and limitations for growth? This is very important when we think about our needs to produce plants for fuel, lumber, and food. So finally, carbon storage and metabolism in plants are critical processes that fuel their growth, resilience, and even reproduction by capturing carbon from the atmosphere and storing it as sugars and starches. Plants provide the foundation for nearly all life on Earth, and as gardeners, farmers, or just plant enthusiasts, Understanding these processes help us appreciate how incredible biology really is. Well, that's it. I hope you enjoyed this video. Thanks for watching. If you like this video, then please like it on my YouTube channel and consider subscribing to my YouTube channel, where you'll find other interesting videos on plant physiology, on viticulture, and on enology winemaking practices. Have a great day.